Let's continue our discussion about proteins and their effect and structure. So it's important for us to remember that amino acids are organic molecules that have both the carboxyl end and the amino end, and that they form these peptide bonds. At the center of the amino acid is an asymmetric carbon atom that we call the alpha carbon or the central carbon. There are four components attached to the alpha carbon: the hydrogen atom, the carboxyl group, the amino group, and the variable R group, or what we often refer to as the side chains. Different R groups give different amino acids their characteristic behaviors. There are 20 amino acids in our body that we need to be aware of. And remember, 9 of those are essential. That is, we have to eat them. We can't make them. Those R groups on the side of that amino acid may be as simple as a hydrogen ion. The amino acid glycine has just the hydrogen atom. Or it may be a carbon skeleton with various functional groups, like in glutamine. They may have ring structures, they may have extended chains, they may have different things attached to them, different components attached to them. The physical and chemical properties of the R group determine the characteristics of that particular amino acid, whether that amino acid is hydrophobic, polar, nonpolar, charged, non-charged. So in the charged or ionized amino acids, at a cellular pH, some of the acidic R groups are negatively charged due to the presence of a carboxyl group. These basic R groups have amino groups that are positive in charge. The basic R groups have amino groups that are positively charged. Note that all amino acids have carboxyl and amino groups. All amino acids have carboxyl and amino groups. The term acidic and basic in this context refers only to the characteristics of the R groups. Again, just kind of as a review, amino acids are joined together with a dehydration reaction, removing a hydroxyl group from the carboxyl end of the amino acid and a hydrogen from the amino group of the other end. Now that peptide bond that's formed there is bond between that nitrogen and that carbon atom. Now, a little terminology that I need to review. At one end of the amino acid is the amino group that we call the N-terminus, and at the other end is the carboxyl group that we call the C-terminus. And that's true of every polypeptide or protein as well. There is an N-terminus and there is a C-terminus to every polypeptide. And again, polypeptides can go from a few monomers to thousands, from a dipeptide, just 2 monomers, to a polypeptide which has thousands. Typically, again, biochemists and physiologists refer to sequences of amino acids up to about 100 amino acids as a polypeptide. And after that, we start talking about proteins. Strictly speaking, though, proteins consist of more than one polypeptide, but that's neither here nor there for this conversation. Now, each polypeptide has a unique linear sequence of amino acids. So there's a unique sequence, like, for example, there's a pentapeptide called famorphamide, which is phenylalanine, arginine, methionine, arginine, phenylalanine. And so that's the sequence of amino acids on there, and each one of them is unique. Frederick Sanger at Cambridge University determined the amino acid sequence of insulin in the 1950s. This is the first time that the sequence of amino acids in a polypeptide was determined. He used protein digesting enzymes and other catalysts to hydrolyze the insulin at specific places. So he break it up into pieces using these enzymes and other catalysts. Then he separated them by a technique that we call chromatography. where you can actually send the pieces through a matrix where they get caught up into different sizes. And so you can use those different sizes to figure out what's in there. Then he used chemical methods after he broke them up into those different sizes to determine the amino acid sequence of those fragments that he separated out. Then he searched for overlapping regions among the pieces to see where they actually joined up. And this took years and years, but eventually he was able to reconstruct the complete primary structure of insulin. Now, to do it, you can do it literally in minutes, as compared to the excuse me, years that it took Sanger. A functional protein consists of one or more polypeptides that have been twisted, folded, and coiled into unique shape. But it's the unique sequence of amino acids that determine the functional conformation for that protein. Now, this functionality of that protein, the final shape of that protein, the final conformation, is reinforced by a variety of bonds between parts of the chain, which is dependent upon the sequence of amino acids. So what that means is the interaction between those R groups that we're talking about that determine the final shape and thus the final function of that protein. So many proteins are globular, just kind of blobs, while others are kind of like fibers. And in almost every case, the function of the protein depends on its ability to recognize and bind to some other molecule. So that unique globular shape or that unique fiber shape determines where or how the function of that protein. So again, heard it before, you'll hear it again, you'll hear it again. Form follows function. So, we talked earlier, and I showed you that image earlier about an antibody matching specifically to the shape of the antigen. So again, shape specific always. So when an enzyme recognizes a specific substrate, it has to be the appropriate shape so that it can match up and bring about that conformational change that results in the reaction. And we talk about signaling proteins. We talked about things that affect how your nerves and muscles work. So we have these natural signal molecules that are called endorphins that will bind to specific receptor proteins on the surface of brain cells, producing an euphoric feeling, producing a relief of painful signals. We have manufactured, because we know the shape, we know the final conformation of those endorphins, we have manufactured drugs like morphine, heroin, and other opiates that mimic these endorphins. So again, once we know the shape, and we do that with a lot of our medicines, once we know the shape of that particular protein, we can manufacture a similarly shaped chemical that will mimic that effect and bring about the reactions that we want. Now, the function of a protein is an emergent property that results from the specific order of the amino acids. So when we talk about emergent properties again, what we mean is a property that has not been seen in any of the precursors to that molecule. So the sequence of amino acids is an emergent property for a protein or a polypeptide that is not seen in the individual amino acids. So when I talk about structure being emergent property, that's really what I'm talking about. So there are 3 levels of structure, really. There's 4. We'll talk about the 4th. But within a single polypeptide, there are only 3. There's primary, secondary, and tertiary structures. The primary structure is the unique sequence of amino acids. So Lysozyme, an enzyme that attacks bacteria, consists of 129 amino acids. The precise primary structure is determined by the genetic information contained in the DNA. So the DNA contains the code for a particular protein or polypeptide, and that sequence of amino acids is contained inside that genome, that genomic structure, that gene for that particular amino acid. When we start talking about, for that particular protein, when we start talking about DNA and genetics and those sorts of things, we'll talk exactly how that's done. But just know that it is your genome, it is your genes, it is your sequence of DNA that determines that primary structure of a protein. Now, even a slight change in the primary structure can affect a protein's conformation. And when it affects that conformation, it affects the function of that protein. So, for example, and I've talked about this before, the substitution of one amino acid, that is valine, for the normal one, which is glutamic acid, at a particular position in the primary structure of hemoglobin, the protein that carries oxygen in your blood. The change of that, the substitution of valine for that glutamic acid, results in sickle cell disease. It's an inherited blood disorder that it's a mutation that occurred in people of African and Mediterranean descent. And so that one change, that one substitution in that one amino acid results in a mal-shaped blood cell which gets caught in the capillaries, which causes pain, and untreated can cause death. Those cells are crystallized and clog your capillaries, resulting in tissue death. And tissue death in the heart is a heart attack. So it's a serious condition, and it's caused by that one mutation. Most proteins have segments of their polypeptide chains repeatedly coiled or folded. So it's not like they're just long chains of these amino acids. So the secondary structure is the coils and folding in the polypeptide chain. And this is the result of hydrogen bonding in many cases. Some cases there are covalent bonds that are affected, but in many cases it's hydrogen bonding between the atom attached to the nitrogen atom and the oxygen atom of the nearby peptide bond. But these hydrogen bonds, when we talked about bonds, that's one of the weaker bonds. And even though they're very weak, you got to realize that they're often proteins that are thousands or even 100,000 peptides long. And so these multiple hydrogen bonds will stabilize the structure of the protein. So there are two things, and I'll show these a little bit later on when we start showing the slides. There are two really typical structures that are formed by secondary structures. Those are alpha helices, which kind of is a coil, kind of a tube-like structure. And there are folds, kind of like you take a sheet of paper and fold it up, that are called beta-pleated sheets. So the structure of silk, which we talked about already, is due to beta-pleated sheets. And so, and I've already mentioned this to you guys, the presence of so many hydrogen bonds makes each silk fiber stronger than a steel strand of the same weight. So then the next structure is tertiary structure. That's determined by interactions among the various R groups. So these include the polar and polar, non-polar, polar or charged areas, possibly charged, negatively charged, ionic bonds between the R groups, and even the hydrophobic interactions and van der Waals interactions among these hydrophobic R groups. So very often, even though these interactions are relatively weak, strong covalent bonds do exist, and one of them that I would ask that you know about is the sulfhydryl groups between two cysteine monomers that hold insulin together. The interaction between the sulfhydryl groups forms what we call a disulfide bond, and that's what keeps the two polypeptides of the insulin protein together. And finally, there's the quaternary structure. This results when you've got multiple polypeptide chains interacting with each other. We'll talk we talked about hemoglobin, and hemoglobin consists of 4 different chains that interact with each other. So the quaternary structure are the 4 different hemoglobin molecules that come together to form that one hemoglobin structure. Another example of that is the collagen. The collagen that has a strong role in our connective tissue consists of 3 polypeptides that are coiled together like a rope. Okay, so that's quaternary structure. So let's go ahead and move on. We're going to talk about each of these a little bit more detail, just so that you kind of internalize them. Primary structure is the sequence of amino acids. Again, it's determined by the sequence of nucleotides in your DNA, in each gene for that particular structure. So here we have an illustration of what we've talked about. Here's the primary structure. We have, again, the amino end and the carboxyl end, and each individual amino acid forms this chain. So it's just a sequence of amino acids. That's the primary structure. Then the secondary structure is these interactions between the hydrogen bonds here, forming either this structure, which we call a helix, an alpha helix, and this structure here, which is a beta pleated sheet. So you see the beta pleated sheet kind of looks like a folded up piece of paper, and the interaction between the bonds of these different groups, hydrogen bonds, and in some cases even covalent bonds, gives this beta pleated sheet very strong nature. And the amino acids here in the helix that forms the alpha helix can form tunnels even that allow some things to pass through them. And we'll talk about that a little bit later on when we talk about the cell membrane. And then the tertiary structure is when you take these things and the sequence of amino acids and put them all together. So you'll have these inside the same polypeptide. You'll have alpha helices, and you'll have beta pleated sheets, and you'll have straight pieces. All these things come together to give this thing the structure. And again, that is highly dependent upon the environment, whether this thing is an acidic or a basic environment, which again is a huge is hugely predicted by the structure of or the environment, the conditions in the environment. And then if I put some of these polypeptides together, this tertiary structure, put them together, we get the quaternary structure. I'm just going to go through that because we talked about that. The coils and folds of the secondary structure, again, hydrogen bonds, repeating, alpha helices, and the beta pleated sheet. Okay, you need to know those. Make sure that you know the difference between an alpha helix and a beta pleated sheet. Make sure that you can recognize them. If I were to put something like a diagram and say, this is an alpha helix and this is the beta pleated sheet. And recognize again that the interaction between them is largely because of hydrogen bonding. The number of hydrogen bonds makes it very strong. Okay, so here's the orb weaving spider here. This web, this silk has beta-pleated sheets. That helps to maintain the long, strong. This is really strong. These help to maintain the shape of the web. These things are made of a different kind of silk. They're elastic. And so what they can do is they can move in the wind and the rain. And even when an insect touches them, it kind of gives, and which makes the insect want to get away. And so as an insect tries to get away because it's so sticky, the radiating strands here are not sticky. It's only the spiral ones that are sticky. Excuse me. And so the spider can walk on the radiating ones, but when some insect hits these sticky ones and it vibrates, it does two things. One, it alerts the spider that it's got some sort of prey captured, and the other thing is it helps to make the prey struggle more. And so it actually helps the spider catch its food. Again, the tertiary structure is determined by the interaction between the R groups. And again, this is very often polar interactions, very often ionic interactions, and in some cases even covalent interactions. They include, again, hydrogen bonds, ionic bonds, hydrophobic interaction, and van der Waals interactions. Covalent bonds here, the ones I was talking about earlier, called disulfide bridges, reinforce the protein structure. And again, important that you remember that disulfide bridge in insulin. It's extremely important for people who have type 1 diabetes. Okay, so here we have the tertiary structure. Again, you may have a straight stretch, you may have an alpha helix, and you may have beta-pleated sheets. All these things come together to give this thing its shape, and it's important that you recognize the role of the environment in determining the final structure. Then I take a bunch of tertiary structures and put them together to make a quaternary structure. That's actually more about a protein rather than the individual polypeptide structure. So here we graphically have what's going on here. I may have a hydrogen bond between two different amino acids across this chain. We have a van der Waals interactions, and remember these two are really fleeting. They don't stick around very long, but there's so many of them. They're made and broken so quick that they add strength. Then you have the ionic bond between two different charged amino acids, and then you may have the disulfide bridge, which again is a covalent interaction, very strong, very hard to break. All right, so here's collagen, which we talked about, which was 3 polypeptide chains that are put together. This is a quaternary structure, and here's the quaternary structure of hemoglobin. I have a gamma, 2 gammas. Is it 2 gammas? Let me think. I'm trying to remember. I know it's 2 alpha chains, maybe beta. Let me think for a second. Let me get my mind together with this. It's 2 betas. Gamma's in the fetus. Yeah. So it's 2 alpha and 2 beta chains that come together and form the functional hemoglobin molecules. Okay, so again, the quaternary structure results when 2 or more polypeptide chains form a huge macromolecule. And again, I showed you previously collagen, 3 polypeptides that are wound around each other like a rope. And hemoglobin, it's a globular protein, consists of 4 polypeptides, 2 alpha and 2 beta chains. Sickle cell disease, talked about that earlier. Substitution of a valine for a glutamic acid. No, that's a glutamine. Valine for a glutamine. Yeah, valine for a glutamine. No, it is glutamic acid, yeah. Substitution of valine for glutamic acid results in a sickle-shaped cell that gets caught inside of the capillary and causes the disease. So if you look here, protein number 6, it's glutamic acid substituted for valine. Here's the change in the secondary and tertiary structures of the molecules. You can see how this has this uneven shape. And so when they come together and form the quaternary structure, it results in this sickle cell shape rather than this normal biconcave shape of a normal red blood cell. So here's micrographs of the two. Here's what a normal biconcave hemoglobin molecule, red blood cell looks like, erythrocyte, and this is the deformed sickle cell shape. So the end result is this thing can't carry as much oxygen, and it gets stuck inside the capillaries, causing those issues. So polypeptide chain can spontaneously arrange itself into a 3D shape as the protein is being synthesized. However, the conformation also depends on the physical and chemical condition of the environment. Alteration in pH, salt concentration, temperature, presence of certain environmental factors, all can cause a protein to unravel, which we call denaturation. What happens is these forces typically will disrupt the hydrogen bonds, the ionic bonds, and the disulfide bridges, causing a protein to become biologically inactive. Most proteins, if you put them in an organic solvent, will become denatured, because what's going to happen is in normal environment for the protein is in a aquatic or in a hydrophilic environment. The hydrophobic amino acids tend to be stuck inside of the protein. So if you put it inside an organic solvent, it's going to flip around and it's become denatured. It's going to expose the hydrophobic regions of that protein to the outside and put the hydrophilic regions on the inside, which makes it non-functional. Before I go on, I want to talk about if I heat something, think about the egg white, which is albumin, which is a protein. It's a structural protein. It's used to build other proteins, and it's kind of used by the developing embryo. But if I heat this up, it's going to disrupt the weak interactions that stabilize conformation as well. And so what happens is I heat that thing up, the proteins begin to arrange themselves in a different shape, and rather than being the shape they're supposed to be, the amino acids, rearrange themselves, and rather than the protein be the shape it's supposed to be, it's going to denature and solidify. Now, in terms of your body, this explains why extremely high fevers can be fatal. Proteins in the blood and in the brain will become denatured by high body temperatures and become non-functional. So that's the reason that temperature typically above 102 becomes really concerning to physicians. Some proteins can return in a process we call renaturation to their normal shape, but others can't, especially inside of a cell. They may be able to do it inside of a petri dish or inside of a flask, but it's really hard to do inside your cell. We now know the amino acid sequence of more than 875,000 proteins and the 3D shape of about 7,000. It's easy to find that sequence because all we have to do is sequence, do what Sanger did, and we can get the sequence of amino acids. The tough part is figuring out how they're folded. And that's kind of what we have to do to make medicines to help when we have some sort of denaturation occur. In fact, most proteins, it's really a sequence of changes in shape before they reach their mature configuration, and they can't do it alone. There are protein molecules called chaperonins. The original chaperonins were called HSP70. HSP70 stands for heat shock protein 70, the 70 meaning 70 kilodaltons. And what happened was that some scientists heat-treated bacteria, and when they heat-treated bacteria, they saw a proliferation of this HSP70 protein, and they kind of tried to figure out what it was. And eventually they figured out that these molecules assisted in refolding, in the proper refolding of the proteins that were denatured by the heat. And so, and they're there all the time. It's not like they're only there when they're heated. They're there when proteins are normally produced. And so rather than calling them heat shock proteins, they started calling them chaperonins. And so the chaperonins, the HSP70s and 50s and others that they found, don't actually specify the final structure of the protein, but they kind of work to segregate and protect the polypeptide while it folds on its own. So the chaperonins, the role of the chaperonin is to protect the protein molecules as they're being shaped and folded. So this is kind of what a chaperonin looks like. This is when it's fully assembled. There's a tunnel going through here. This again is a hollow cylinder. These are lots and lots of amino acids that form this tube-like structure with a cap on it. And as the polypeptide is produced, it enters, the cap is removed, and it enters in through the cap, and then the cap is replaced. Okay, so that causes the inside of this thing to change shape. So if you look here, you see how this is represented. It's not really this way, but it's a good representation. There's a straight tube, the polypeptide enters, the cap attaches, the tube changes shape, and so it creates this hydrophilic environment for the folding of the polypeptide. There's some speculation it actually plays a role in helping it fold. And once the polypeptide is matured, we call it a matured polypeptide, the cap is once again removed and the protein is released to perform its duties. At present, we're using X-ray crystallography to determine the structure of a protein. And so what has to be done is the protein has to be crystallized, which is a very complex, even today, a very complex sequence of reactions. And so what happens is the X-ray, that crystal is X-rayed, and then the pattern that is produced by the interaction of the X-rays with the protein is captured by a computer, and that computer then builds a model of the structure based upon that diffractory pattern. It's a really complicated process, and thank God for computers because it makes it possible. Another method that we use to determine the structure is something called nuclear magnetic resonance, or NMR spectroscopy. We do not have to crystallize the protein. It's a native conformation of the protein. What happens is there's a huge magnet. It's kind of like when you take an MRI. There's a huge magnet. It switches polarity from one to the other, and as it does so, it changes the orientation of the electrons in the atom, in the structure, and then again a computer will interpret the information to produce a structure of that protein. Of the two, because we don't have to crystallize it because it's a native conformation, NMR is much more becoming much more widely accepted. The third mechanism to determine protein structure is bioinformatics. Bioinformatics predicts protein structure from amino acid sequences. It's a very complex. A computer does it. We do the Sanger sequencing of the protein. We figure out the sequence of amino acids, and once that's done, it's fed into a computer, and the computer looks at possible interactions. When you use bioinformatics, you don't get a single structure. You get multiple structures. Typically, after bioinformatics, people will use either X-ray crystallography or NMR to determine the final structure. So if we look here, this is X-ray crystallography. We make the crystal. There's an X-ray source. We generate an X-rays, send the X-rays through the crystal. The X-rays are diffracted, and we have this digital detector, and this is what the end result looks like. Again, you have to be well skilled in how to do this to interpret it. And so from this, they determine this. And again, this is all done by computers, not by people looking at it. Okay? Next extra will be nucleic acids.